What's up, AP Penguins, and I guess recappers? I'm not really sure what we're calling the APsolute recap students, but welcome to today's live. We're doing Unit 5 and Unit 6. So in just a moment, Melanie is going to join me. So welcome to everyone. So Unit 5 is all about genetics and meiosis, and so we already did the meiosis last, and so today we're going to hit the genetic side of it, and then also we will get into Unit 6, which is all about transcription and translation, mutations, gene regulation, and hey, there's Melanie.
Hi,
welcome, Melanie.
Happy Sunday.
Yes, why did we think of 8 p.m. on a Sunday? I'm not really sure. So before we get too deep, because we always forget to do this, hi. I'm Miss Jones, or Tiffany, from AP Bio Penguins, and I teach in Georgia. And you are?
I'm Melanie King. I am from the APsolute RecAP. I teach in Pennsylvania.
Woohoo! So how are you doing tonight? You doing good?
I'm good. Yeah, my students just finished up their Unit 8, so we're about to start reviewing. It's so close. It's so close, right?
It is. So students, as you're getting in, hi, welcome. Don't forget any questions that you have about Unit 5 or Unit 6. You want to put them into that little question box. Like there's a little, it looks like a little comment thing, and then you'll put question mark there.
It reminds me of the question mark box from like Mario, right? That little,
yeah.
like that little square.
Yeah. So that's how we're going to see your questions. So go ahead and you'll put them down there. If you put them in the chat, we don't really see them because it kind of moves up so quickly sometimes. So make sure that you have any questions on Unit 5 or Unit 6, that you put them down there. There hasn't really been a lot of questions that were submitted ahead of time. I think there was one. Right. There's one question.
So if I know the numbers can sometimes be confusing. So if you're here, you're like, well, I don't know what Unit 5 is. My teacher's never said the words Unit 5. That's okay. So Unit 5 is about heredity. Unit 6 is gene expression and regulation. They usually fall in like the meat and potatoes middle of the year for most teachers, regardless of how it flips. So some courses tend to start with like evolution and ecology, but then you hit this right about the middle of the year, or you start in the beginning. Either way, thank you for joining us.
Yes. Okay. So let's just go ahead and jump into this. So the first question we have, and I'm going to repeat the questions. At least I try to remember to do that.
Right.
Because when we play the live back over on YouTube, those questions don't show up later. So, and sometimes Melanie doesn't see the questions. So I'm going to go ahead and just say the question. So how does RNA splicing lead to increased variation? So first thing, what is RNA splicing? Melanie?
Well, it kind of self-describes itself, right? To splice, to cut, to biology tends to give the answers away. It's like reading Jeopardy questions. So we're gonna have the answers in there somewhere. So RNA splicing is going to be the way in which we have the relationship to, I'll say, introns and exons in our RNA molecule, but we're specifically referencing eukaryotic cells, and that's important to indicate why don't we see RNA splicing in prokaryotes, Tiffany?
Well, we don't see it in prokaryotes for two purposes. Number one, there's no introns in a prokaryotic genome. And so they don't really have that purpose to cut something out. And the second reason is because of the fact that there is no nuclear envelope. And so as soon as you start transcribing it, and as soon as you start reading that DNA to make the mRNA, a ribosome is able to attach to it because there's nothing to separate it. And then it starts the process of translation. And so if there was an intron in there, then it would cause issues. So those of you who don't know what an intron is, because we just said that a couple times.
Oh, yeah.
Introns are non-expressed information. So I always tell my students that it's like in between, and that the exons are the expressed or the exciting information. So I always say
introns are interrupting.
Right.
Now, it doesn't mean that introns couldn't have important information, just not for this particular gene to be expressed. And that's going to be important to know because that relates back to the question. So I'll let you keep going.
So how does it cause increased variation? So due to which parts of the mRNA is remaining in there is what's going to determine what protein is made. And so sometimes those exons can be cut out, which will give us a different mRNA. And that's actually, let's make a direct connection. That's actually how we get the different glycoproteins on your B cells and your T cells is by that alternative RNA splicing. And that's how we get that variation, is because of the fact that even though we have the exact same genome and we have the exact same DNA that's making for those receptors, due to alternative splicing, you can make different proteins or different glycoproteins from that. I think I went on tangent, sorry.
So we end up with like differentiation, right? And so differentiation is so important when it comes down to cell specialization and cell function, especially with the immune system, which you just gave a great example of. And one thing to note about RNA splicing, so since we don't see it in prokaryotes, you know, that origin of process is going to be distinct to eukaryotes. It is a common ancestry feature of all the eukaryotes.
Right. And if we want to talk about the regulation that's occurring within a prokaryote, that would lead us into the Operons, which are a whole can of worms in themselves.
They are.
There was something I was going to say. Oh, so I just mentioned a connection to the immune system. I want to make sure that I kind of refresh your mind for a second. The immune system is not tested on the AP exam. So there are no animal systems on the exam. There's no endocrine, there's no immune, and there's no nervous. Although those three were in there as of 2 years ago and forward up to, I think, 2013. But they can be used as illustrative examples. So they can describe to you and tell you how the immune system has these different receptors. And then you would have to make the connection to, oh, it's alternative RNA splicing. And that's how you got the variation. But they would have to give you everything you need as a background to be able to make that connection or to make that leap.
And so it's really important, I think, that when we get to this stage of studying, we start learning in context. So it's it's like almost like exposure therapy, like expose yourself to as many concrete examples of this applied information as possible. If you don't know where to look, download the beast that Miss Jones over here has put up there for you. Tons of illustrative examples that are released AP questions. Go get it on our Weebly.
Yes. And somebody, since we're already talking about the RNA splicing, just real quick, remember that it is done by these things called spliceosomes. So there's a bunch of SNRPs,
SNRPs.
Yeah, I call them SNRPs. What do you call them?
I call them SNRPs. Yeah.
Okay. SNRP.
PS.
Yeah. And so what they do is they bind to a specific sequence at the end of each of the introns, and then they fold together to take that out, and then they connect back the two exons. You don't really need to know that in-depth component of how it happens. You just have to know that when we go through RNA splicing, that we cut out those introns, and then with alternative RNA splicing, we cut out sometimes some of the exons, which gives us that different mRNA. So that was answering the question that was real quick in the chat with that. Oh, we were up to lots of questions.
Okay.
Hey.
Are we gonna die? Are we gonna bounce back? You want to just pop into 5 and 6 wherever they fall, or do you want to try to go sequentially?
Yeah, let's just jump around. You know how we like to just do whatever. You literally have no idea what the intron is. Can we start from the very beginning? I think we did that though. I think they got that.
Let me back up just a little bit. So introns are going to be sequences of nucleotides in an mRNA transcript. Okay, so when you have your DNA code transcribed into mRNA, then translated into protein. So you're going to need to make sure that mRNA transcript is exactly what you want it to be before you go and translate. So when we say intron, if I zoom in and see what am I actually looking at, you're looking at a sequence of nucleotides. Okay, you're looking at your A's, T's, C's. Nope, I'm in mRNA. Your A's, U's, C's, and G's in whatever order they happen to be, in order to then go into translation. So you're in the intermediate step of the central dogma.
Okay. So let's jump into, I guess, those operons. Yay, operons. So can you describe the promoters and operons and their functions? Okay, so first thing, what is an operon? So an operon has three main components to it, right? You have the operator, you have the promoter, and then you have the genes that it controls. Now remember, operons only occur in prokaryotes, okay? And so I like to think of the operator as this location where a big rock can bind, okay? Like, so imagine we're looking at railroad tracks, and there's a spot where the big rock can bind. So the operon is where the repressor is going to bind. So the repressor binds to the operator, which can either turn the whole operon on or off. Okay, so the repressor is your big rock. So if the big rock repressor binds to the operator, RNA polymerase is unable to get to the promoter. And
there's no trains going by. We're on the train tracks, right? There's no
the train
trains,
is RNA
right?
polymerase. And so if RNA polymerase, my train, can't get onto the tracks, it can't read the genes or can't go down the train tracks. And so it all comes back down to, is your repressor on? If your repressor is on, your operon is off. If your repressor is off, your operon is on. And that's when you have to think about the inducible versus the repressible operons. Melanie, can you take it away with the repressible versus inducible?
Sure. So it comes down to what product are you making? So if our, wait, let me get your hands right. So if our, wait, does your son have any trains nearby? I feel like we should have props for this.
Oh, not nearby. Sorry. Those
not
are
nearby.
All downstairs.
So it comes down to what product are we making? So the lac operon is probably the one that gets the most attention. Would you argue that that's a fair statement?
Lac operon and trip operon definitely are the ones that get the most attention, which means it's probably are not
won't be
on
the exam.
Probably not.
So
especially on the digital exam that is able to be, gotta protect all content. So
I just had a poll. Sorry, I'm getting distracted, but I just had a poll on my page about how many people were taking the digital exam, and it was about 50%.
So
it was pretty high. Are you students split between?
Oh, I have a lot of students that are all going to go virtual. So who knows? Okay. I only recommend the paper, but that's my own personal opinion.
All right, so
back to operons. Go.
Yeah, let's remember. So the lac operon, that's going to be inducible. Am I remember that correctly? Or repressible? I can't remember.
Inducible operon.
All right, I'm gonna muddle it up. But lac, named after that, it's going to be working with lactose. So when way I look at these diagrams, it comes down to efficiency, right? Do we want to produce a product to break down lactose if lactose isn't around, right? So when you have lactose present and lactose binds to my repressor, it's going to remove it. Am I saying this correctly?
If you have lactose present, it's going to cause your repressor to turn off, so it's not going to bind to the operator anymore. So your repressor is off, your operon is
operon's on.
Notice that my eyes roll up because I have to think about this too, children. Like, y'all keep in mind that we as teachers still have to conceptually think through things. It's not that we just know it. We think through it in the same logic y'all do.
In my head, I'm seeing a lactose molecule binding in an exact fit, pulling something off, and then having expression. So I've said this a million times, the irony, right? I need to draw things. So even doing Instagram lives, to be honest, like for me, like this is all coming from our heads. I'm trying to see things. It's important for us to look at diagrams too.
Okay, so our repressor is turned off because of the fact that we have that lactose binding to it. And so we then are going to make the materials that will allow us to bring in the lactose and then also to break down that lactose because we have to get the glucose from the lactose. And then versus the trip operon is going to be a repressible operon. The job is to turn it off if trip is present, because why am I going to make something if it's already there? And so I think we know the E. coli lives in our gut, right? So whenever you eat something, of course, there is tryptophan in your diet. And so why would you need to have the bacteria try to make it if it's already there? And so if the trip is present, then it's going to bind the repressor to turn the repressor on, which turns your operon off. The end.
I like the hand signals. And do that, right? You could be in the middle of the exam. Like, who cares what you look like? Use your hands, right?
Oh, yeah.
I'm always whenever I'm testing I know it's a different unit, cellular respiration, I always see the students with their hands in front of their face because they're breathing on their hands, trying to remember the products of cellular respiration and making sure they're in the right spot.
And if the physics students can make their XYZs in their hands, so can we do crazy things. Definitely. We can do the mitosis dance during the exam.
Do it.
Okay. Sorry. I think we did that question, so let's go back to find some more questions for us.
So if you're just joining us, we are doing units 5 and 6. That would be heredity and gene expression.
Okay, so let's go through the process of transcription. So what is the process? What happens during transcription? So number one, where does transcription occur?
Well, it depends on what you are.
Okay, let's do them both.
Right. So transcription is going to occur where your DNA is. So you're going to be in the nucleus if you are eukaryotic. You're going to be nucleoid is going to be your prokaryotic term, which is the location where your DNA is for a prokaryotic cell. I like to think I'm a big Latin person, so transcription has the root word scribere, which is like scribe. So when you're a scribe or you're writing down a transcript, you're still in the same language.
You use the same example.
No way.
Yes. Talking about the same language. Okay, yeah. So you keep going. Sorry.
Okay. So, there must be a reason. There must be a reason. Okay, so you're still in the same language. So for transcription, the goal, go from DNA into RNA, but we are still speaking nucleic acids. Now we have to remember that RNA is not going to have thymine, right? We're going to be substituting our uracils, but in terms of our association and how we get there, it's going to involve a series of enzymes, and there's only a few of them that you have to know, but there can maybe others that are referenced on diagrams or on, you know, different prompts. Have you seen a lot of enzymes in FRQs in the past? Tiffany's read a ton more FRQs than I have.
Not since the redesign.
Not since the redesign. Okay.
No, not so, when I say not since the redesign, I mean like not since the 2012 redesign. Pre-2012, there was tons of them.
A lot of information.
Yeah, because those were more of the rote memorization exams. Now we're more application-based. So I haven't really seen a lot of it since then. It's more, it'll probably say, you know, maybe that the enzyme required for transcription has been inhibited. What would happen? And you make some applications from that.
Okay.
Sorry.
That's all right.
Okay. Anyway, so there are 3 steps that we go through with transcription. The first step would be initiation. This is where we're going to find our, at our promoter site, we see the RNA polymerase will bind. And I have another crazy example because
let's do it. Before you get into it, just for those of us listening, there are going to be a lot of analogous things happening to replication. So, and that's not accidental. So Tiffany said at a promoter region, that's important because we want to transcribe a specific sequence, whereas in replication, we would be copying the entire genome. Go ahead.
So it's like a baseball mitt. When you have your baseball mitt and you want to catch a ball, you don't just catch it in the mitt. You don't just catch it barehanded. As a baseball or softball player, you would catch it with 2 hands. Ball comes up, right. You catch it with your other hand. And so we don't just have RNA polymerase binding to the promoter site. We also have a lot of these other transcription factors that are going to kind of make that nice secure fit to allow for the initiation to start. Once it gets started, everything else falls off, and it's just RNA polymerase binding down. And as it goes nucleotide by nucleotide, it is able to actually open up. It being RNA polymerase can break those hydrogen bonds to open about, I think, like 5 to 15 nucleotides.
Yeah, something in that range. It's not very wide.
Not important.
It shouldn't be very wide,
no.
Just from the nature of protecting the sequence.
Right. And so that's when we'll see the base pairing, you know, A goes to U, T goes to A, C goes to G, G goes to C. And that's the elongation phase where it just kind of keeps going. And depending on if we're looking at prokaryotes versus eukaryotes, prokaryotes have a terminator sequence. So they get through the terminator sequence, and that's when RNA polymerase falls off. Versus prokaryotes, it just kind of goes until it falls off. It usually goes through this polyadenylation sequence, which is basically a whole bunch of A's or adenines. But it just falls off of the sequence.
So in this, yeah, go ahead.
So much stuff happens in post-transcription that we're able to get rid of all the extra stuff.
So we have to remember that DNA has directionality when we're working through transcription.
Oh, good point.
Right? And so just as when we were going through replication, DNA polymerase had, and polymerase means make a big molecule, like make a polymer. So
RNA polymerase makes an RNA polymer. DNA polymerase makes a DNA polymer. The enzymes tell you what they do. Helicase breaks the helix.
Yeah. Well, okay. Well, so topoisomerase. I don't know the root for that. Do you know why it's called topoisomerase?
You have to be. Why do you do this to me?
Because
all I have is a way to remember it, woman.
I'm sorry. Okay, we're gonna find out, and we'll post it on one of our pages.
I think what is the origin? Because it breaks and makes into like an isomer, and it's to unravel. But I don't know why. It just makes me think like a top, like it's unscrewing like a top.
I like that answer. Okay, so be mindful of how you express the movement of the polymerase. So it's going to synthesize 5 to 3. It's going to read 3 to 5. Okay, but don't just say it goes, because that tells us nothing. Okay,
readers won't give you credit for it because that's vague statements.
Yeah, take out your pronouns, put in your proper nouns, and your terminology, because you want to earn the points where you can. And 5, 5' and 3', we're referencing, if you can't recall, right? So we're referencing the the nucleotide, the nucleotide monomer itself, right? So a nucleotide made up of pentose sugar, made up of nitrogenous base, and made up of the phosphate group, right? So my phosphate group side of the nucleotide monomer, that's going to be on the 5' side. I'm referencing the number carbon in my pentose. Whereas the 3' side is going to be on the not bound to the phosphate, right? I need to draw it. I can't help
Third
it.
carbon. So when you count around,
carbon.
so you start at the oxygen and you count all around, and you start with your nitrogenous bases, that's number 1, then you have 2 carbon, and your third carbon is where you'll see basically the hydroxyl of the sugar is where 3 is, 4, and then 5 is where the branches, and then off of the branch is where your phosphate is.
Yeah, the
Not
5th carbon.
To know these numbers. Like, just know 5' is phosphate, 3' is hydroxyl. If you need to help remember, just remember poo, POH, 5 to 3. I was laughing because my second year teaching, they looked at me and go, Miss Jones, it's poo. And I'm like, oh, God. Poo. Oh, gosh.
The 2' carbon, since we're talking about pentose rings, that's going to be one of your distinguishing features between ribose and deoxyribose too.
Right, because on the second carbon is where you're going to be missing either one oxygen or you'll have the oxygen. And deoxyribose is only missing that one extra oxygen. And so they can actually give you an application question in that way, in which they can give you the numbers of carbons and oxygens and hydrogens. And then they can ask you about, well, what kind of structure would this be? Or they could give you the number of adenines, thymines, uracils, and all that, and then ask you something from there. So keep in mind what the structure of these things look like. And I know we just kind of went off on our own little tangent, but this is what happens with us. That's why we still have 22 people joining us. Sorry, y'all. We do this. This is just us.
All right. So heredity, gene expression, and
ooh, somebody's even answering our questions in the question box.
Oh, like it. Post them sequentially. Let's make it happen.
I know. Okay, so this is a really quick answer, and I think we already said it, but I want to make sure that I answer and we say it and we're clear on this. DNA polymerase is used with replication. RNA polymerase is going to be used in transcription. Now, we do make a little section of RNA when we go through replication. That's a primase, which makes the RNA primer, and it does get replaced later on by DNA polymerase. Don't ask me which number. You don't need to know which number.
2, 3,
just know
so
DNA polymerase in life is good. And we have discovered that I'm not very good at balancing this whole question box thing.
So when is what Tiffany just answered. Where is what I'm going to answer. They both function in the same place. Okay, so DNA polymerase and RNA polymerase are both going to be an important role inside the nucleus or at the nucleoid region. Ratios. Oh,
I'm sorry. I was letting you finish. I just paused the question while I wait.
No, okay. Okay, wait to remember to read the question.
Right. So what ratio should we try to remember for the Punnett squares? Definitely know a monohybrid cross is a 3 to 1. Definitely know that a dihybrid cross is a 9 to 3 to 3 to 1. And know that a dihybrid test cross is going to be a 1 to 1 to 1 to 1. Those are the common ones to know. If you're talking about independent assortment, then a monohybrid would be 1 to 2 to 1. Same thing with codominance. But from that, you're probably going to have to do the math yourself. And I don't know, like we're supposed to be doing genetics in this too. So
I'm not a huge fan. I mean, obviously there's some numbers that get stuck in your head. I mean, 9 to 3 to 3 to 1, you can't help it. I'm not a huge fan of memorizing the crosses, because here's the scoop. Like it would be a real shame if you do the work and then don't answer the question, right? You get wrapped up in the numbers and then never actually address the prompt, which may not have been asking you for ratios.
Right. And I will tell you, speaking of ratios, there was a free response question a couple years back in which it did ask you the ratio, and the students wrote just the ratio, and they did not make it a complete sentence, and it did not get counted. So remember, you must write complete sentences. So if they ask you for a phenotypic or genotypic ratio, usually it's going to be phenotypic, you need to say the ratio is. Those 3 words right there can be the difference between 1 point or 0 points. And I always tell you, make it a complete sentence every single time, no matter what.
And identify what the number is referencing, because they don't want to assume that you are, I mean, you are likely identifying them as dominant to recessive, and just because your teacher has always expressed that and they know what you mean, you don't want to assume that the readers, know anything of what you might mean.
We can only grade what we see. So if it's not on the paper, we can't give you credit for it. Same thing as if you slip up a word. So somebody today in the Facebook teacher group, clearly in the sentence that the student had written, you can tell that they meant to say female instead of male. You can clearly tell it, okay? And then they fix themselves later on in the response. But that is not a point, because number one, you contradicted yourself because you first said male, then said female. And number two, even though you completely described it, you said the wrong word, which means you might not understand.
Even if you did the work to back up the answer, you said the answer wrong in the end. And the question real quick says that we're directly on this. It says, should I do 3 to 1 purple to white, or should I do 3 purple to 1 white? It should be 3 purple to 1 white. And speaking of that ratio, make sure you understand what that means. That means 3/4 are purple and 1/4 are white. I know that some students will think that means a 1/3. That's what, if you write a 1.3, some students thought that meant 1/3 instead of meaning a 1/4 and 3/4. Right, with the colon. Do they accept fractions too?
Yes. As long as it is a correct ratio that you've given, it is correct. That year we were allowed to take 1/4 to 1/4 to 1/4 to 1/4, 1 1 1 1, 4 4 4 4 4, and I hope you know that I'm not counting my numbers in my head, but you get the idea. I guess 16, no, that wouldn't have worked. Never mind.
So while we're on Punnett squares, because you don't want to do work you don't have to do. So if it's a dihybrid cross, it's almost very unlikely you'd have a 4x4 when you finally get your gametes. It's like incredibly unlikely you'd have a full 16 box cross. And I know you actually encourage your students to not do it that way, correct? Yeah.
So with it, my recommendation is always do your small 2x2s and then use your probabilities, right? Do the cross of the first letter. So let's assume we're doing A's and B's. You do the A's on one Punnett square and the B's on the second Punnett square. And then if we were looking for, I don't know, the heterozygote from that, that would be 1/2 of them and 1/2 of them. So 1/2 and 1/2, 1/4. Well, that was not right, but yeah, it is. Go on.
Yeah, you're good. You're good. And I think what it comes down to is consistency. So what you want to do is find your groove, continue your groove through all the problems that you practice. And I know it can be really tempting to just go, oh yeah, I know it, and move on. But even though it'll take 20 extra seconds to practice writing that full sentence answer at the end of the prompt, it's really important that you do that. I really want to make like a jokey pun based on this question. I can't think of one.
You want to make a jokey pun on this one?
Pun. Well, I don't have it, but
while she's thinking of her joke, what is the difference between Mendelian genetics and non-Mendelian genetics? So first thing,
3 letters. Non.
Oh my god. Oh my god. Anyways, Mendel lucked out. Like, we can just totally say that Mendel just straight up lucked out, right? Those peas on that monastery, and the traits he chose to follow, they went through complete dominance, in which it was either this or that. There was no funky characteristics. There was no incomplete. There was no codominance. There wasn't epistasis. There wasn't anything else in the world.
No. You could have grown anything else and it would have been...
Yeah, it wouldn't work. Okay. So Mendelian genetics is literally just looking at those complete dominance traits. Versus non-Mendelian is looking for anything that violates that. So incomplete dominance, codominance, and all that. But I believe if you look in the CED, I believe they've actually put all the codominance and incompletes all with Mendelian. And the non-Mendelian is talking about your chromosomal, isn't it?
Let me look. I have it right here. I will come back and answer that in just a second.
Yeah, I'm just gonna talk. Just openly
keep going.
Talk.
I'll look it up.
So let me just go over these different types of dominance I just mentioned. So complete dominance has to do with that if you have at least one dominant allele, that it's going to be able to make enough of the product that the homozygous dominant and the heterozygous appear the same. And so things to remember about this is that in all of this, we're looking at a protein product, right? And so usually your recessive trait is going to have a non-functional protein, or it's not going to make any protein. There'll be just no protein made. And that's why whenever we see a homozygous recessive, that's why you can finally see that trait. It has nothing to do with that the dominant's fighting the recessive. It just happens that the recessive doesn't make anything, so there's nothing to cover that not making. With incomplete dominance, we're going to see that the two are dominant, but neither one can actually cover the other, so they both make a product. And so if we're making a red pigment and making a white pigment at the same time, those two pigments are going to blend together. And that's why you can see the red snapdragons, the pink snapdragons, and the white snapdragons, because the pink is the co-dominance, showing the blend of the red and the white allele, or the red and the white trait. Co-dominance is looking at that we have both of them are dominant, and you can see both of them independently. So that would be like your blood type. You have the A blood and the B blood type, and if you look at a cross between those, you could have both the A glycoprotein and the B glycoprotein. Actually, I think it's a glycolipid, no matter, on the actual red blood cells. So you can see both of them at once. I think it's a glycolipid. It's not important. And epistasis isn't actually in the curriculum anymore, but it has to do with where one gene covers another gene. Now, did you find out the answer to our question?
I actually didn't, because those are all like specific examples. In Mendelian genetics, it's not in Mendelian genetics, I can tell you that.
Okay. So
but
maybe
in
I was
non-Mendelian
right.
genetics, the only example that they really give you are sex-linked.
Thank you, Samantha. Samantha looked it up, and it's a glycoprotein. Thank you, Samantha.
Thanks, Samantha.
That's what Kaufman usually does, all this data checking.
Yeah,
where is she doing today? Okay. Let's see. Okay. Anyways, also speaking about, yes.
Oh, no, go ahead. So what I was in the CED when you were talking. So I don't know if you said this already. I apologize if it's repetitive. So it's really important with non-Mendelian genetic problems that you interpret your Punnett squares appropriately, since they are going to follow some non-traditional patterns. Did you say that?
Yes.
In particular, if we're talking about sex-linked traits, please, please, please use X's and Y's in order to follow the pattern and interpret it as it needs to be later on. And not all organisms use X's and Y's, so they may throw. And not all males are X, or you know what am I trying to say?
Males assigned at birth.
Thank you. But it's going to be varying combinations of chromosomes that you might get in different species. So whereas females are have two X's, that may not be the case in other species.
And that actually was a free response question 3 years ago. It was a DW. And in certain environments, they had temperature that could cause a genotypically female to be phenotypically male. And so they asked you to find on the Punnett square what was going to happen. And so I believe it's maybe the 2018 exam. If you want to go and check me later on life, it's definitely in my beautiful 245 page.
Yeah.
Beautiful thing. So definitely unit. Actually, it would probably be an evolution, because there was a question that connects to evolution in there. But so definitely in there. Is someone really writing that long of a question to us? Oh my God.
3 colors of snapdragons. Someone doing their homework?
This question is on the review, and it's not possible. There are 3 colors of snapdragons. Solve for all values if there's 100 red flowers, 800 pink flowers, and 100 white flowers. Yeah, but that's Hardy-Weinberg, which is unit 7, which we're not doing unit 7 tonight. 69 is nice. Can you please just send me a message later, and I will answer that in my stories, since I'm currently doing the unit 7 in my stories. I have no problem doing that Hardy-Weinberg problem for you. But this live is for unit 5 and 6. Okay, so we answered our genetics question. Should we talk about the mitochondrial, or are we good with that?
Yeah, I mean, no, might as well. I don't think we'll get a question about that in the chat box. So be mindful of the fact that because mitochondria and your plastids, right? So those would be your chloroplasts, your leucoplasts. They do have their own DNA. They were formed through endosymbiosis, and as a consequence of that, we may get expression of those traits, and you can trace them through pedigrees. So although it may be unlikely you'll see like a Punnett square problem with those particular traits, definitely could be something that pops up in pedigrees. And so your mitochondria according to the CED, are going to be passed on through egg. So those are going to come from mom, and you're going to find that maternal lineage will be present for mitochondrial expression. Okay, sorry. Yeah.
You're good. I know we already answered this, but somebody brought this up in the chat, in the little question box, and it was after we had said it. So I want to make sure that we mention this, so they're clearing up their confusion, because they're confused. The directionality during transcription. Anti-parallel. Okay.
Anti-parallel, right? So my hands are pointing in opposite directions, but they are not intersecting. So DNA is going to have a 5' to 3' side, and then the opposing strand, what's happening? The opposing strand. This is the length that we go to. Is that a Sharpie?
Yeah, of course it's a Sharpie. What would you write on your milk with?
I don't know, but I love it. All right, so 5' to 3'. So there we go. Pointing in opposite directions. This essentially means that the molecule has a phosphate ending on one side, on the 5', and then it's going to end in the hydroxyl of the sugar on the other side. So when we talk about whether it's replication, DNA replication, or transcription, when we talk about DNA splitting and RNA polymerase binding to DNA, it's going to bind and read 3 to 5 and synthesize new nucleotides of mRNA 5 to 3. It is backwards. It's a mirror image, but I think we get the general idea. Are you going to try and write it backwards so that it comes up on the screen forwards? I love it. All right. So directionality is going to be important when you are expressing your answers. It's also going to be important when you extract information from a diagram. And just like we talked about with pedigrees, giving your full expression of the answer, using your nouns appropriately. Okay.
So let's just take a pause, because there's a lot of questions in the box about the exam and about administration. So I just want to make sure that we, everyone's voices are heard, but we do try to focus on 5 and 6, and there's not really any more 5 and 6 to go.
So if you haven't heard, there are 3 exam dates.
I hope you've heard. The first one is all paper. The second one is digital, and the third one's digital. The first one is on the 14th of May. The 27th of May is the second one, and June 11th is the third one. It's digital exam. Which administration do you think is best to take, Melanie?
I think it's best to take whichever version you're preparing for. Okay. And I mean that with most intention, right? So content-wise, the College Board is doing their absolute best efforts and to make them consistent, right? To make them equally equitable. Process-wise, equally equitable? It's a little redundant, right?
Equitable.
Equitable. Process-wise, they are different. And so if you're taking a digital exam, you need to change how you're preparing yesterday, because you want to give yourself the same restrictive parameters that will exist for you on the digital exam day. Now, let's talk about paper first, because paper is the OG version, and because of the redesign, it's the first version of the exam that actually has been given. So how does the paper exam work, Tiffany?
What do you mean paper? You can go forward, backward. You're in multiple choice question. You have 90 minutes to do 60 questions. There's a combination of short questions. Independent questions, and then there are these set questions that will have a long prompt, and then you have questions to go with those with the sets. If you look in the CED, it tells you the percent that's going to be from each unit. The most heavily weighted units are Unit 7, which is natural selection or evolution. I don't know what the actual CED calls it. And then Unit 3, which is the cellular energetic stuff. So cell respiration, photosynthesis, enzymes. And when I say it has a higher percent, I mean like literally there's probably 1 or 2 more questions in the other sections. They do a great job making them relatively equitable. The benefit of doing the paper exam is that you're able to, yes, this paper exam is different than any other year. This is the first year of this redesign. So The advantage of the paper exam is that you can go back. So I always tell students, if you don't know the answer, you can softly bubble in a guess. And then if you have time, you can come back to that question. With the digital exam, you're not going to be able to go back. So you want to spend maximum time on each question. And so that time, I think, is about, what, 1.5 minutes per question?
About 90 seconds.
Yeah. So you probably want to spend less time on the singlet questions and more time on the set questions. When I did the math on the practice exams, there was approximately equal amounts of set questions versus independent questions. I don't remember. I've kind of gone on a tangent here.
I mean, I think like the what it comes down to, you really don't have a ton of time to hem and haw, right? To think about the answer, put the answer down, and move forward. Advantage to the paper design is that you will have the ability to move back and forth and jump around should you need to. You really don't have time to, but you at least have the opportunity to check your work if you have paced yourself properly, that you can go back to some of those questions maybe you were a little like hesitant on for that. And then the FRQ is going to be styled how?
You'll have 2 long free responses and 4 short free responses. Same thing for the digital exam. And then you can go, forward, back, wherever you want. I always tell students that renumber them yourself. Go through, read them all, figure out which ones you know the most, and answer what you know first, and then go back to what you don't know last. Digital exam, just start with the first one, set a timer for 25 minutes, and when that timer goes off, move on. If you are done before that and you have looked over and there's nothing else you can add, and you've guessed everywhere you can guess, move on. Use that extra time in other sections. You should have about 25 minutes for your first question, 25 minutes for your second question, and then 10 minutes for questions 3 through 6. And that will be the maximum 90 minutes that you get for your responses.
And if you're doing your same will be true for multiple choice, you can only go forward. If you haven't downloaded the digital software yet, download it, check it out.
I haven't even pulled up on my computer to show you, just in case.
Yeah, do you have it?
I haven't pulled up, but there's no exam in it.
I mean, that's fine.
Can I flip my screen?
Yeah, you can flip your screen. So play around with it. You can interact with the program pretty well, but there are some features that are a little different from if you took exams last year digitally. So everything has to be done in the program. There's not going to be any uploading of answers. There's not going to be any handwritten answers. And that's going to change a couple of things for biology specifically. 2, 5, what?
Questions 2 and 5 will be different. So if you open up the CED at the very, very back of it, and I keep referencing the CED, y'all, that just is our curriculum. It's very open book. Like, you can literally know exactly what is in the curriculum. Just search AP Bio CED and you will see what your teachers are teaching you from. But if you flip to the back of it, very, very end of it, there is a formula of what your first responses will look like. It's going to look like this no matter what. The difference is that on question 2, you don't graph anything. They will assess that skill differently. And then question 5, there's usually a diagram that you're going to have to manipulate somehow. On the practice, it was like a pedigree, and you had to label the genotypes of different organisms. I can't remember what it was on the other ones. I haven't focused that in depth on the practice exams because I try not to look at them, so I don't tell my students anything. Plus, I don't want to get in trouble from College Board by saying something on here about it, which is going to come into play really problematic when we get to the later parts after I do look at them. Yeah, we don't want y'all. We can get
like our
trouble.
Jobs.
Yeah, we do. We can get in trouble for sharing things we're not supposed to share. Somebody asked, how was the curriculum redesigned? So we're going to answer this, and I'll jump back into our question box, see if there's any more that's been jumped or asked. The redesign for us wasn't really an overhaul redesign. They took out the animal systems. They took out a little bit on plants, not really much. The big overhaul for us was just that they separated the science practices from the knowledge sets. So if you look in the CED, any of those science practices can be paired with any of the knowledge sets. And back in the day, they used to be directly tied to each other. So you knew exactly how things were going to be assessed. Now it's open book. You can do whatever you want.
Yeah, I think,
not open book, but
I mean, not literally open book, but it gave the teachers a little bit more latitude with how they taught, since they were really trying to enforce skills that would be transcending content, right? So if we think about our role in teaching AP biology, it's not really for the biology. I mean, I'll say it is for the biology, but it's not. we want to make sure that you have skills beyond this class. In fact, if you get a 2 on the exam, you know, you still learned how to take this type of a course, and that is going to be significantly more valuable to you than any score you get in May or June. I just need to say that, get off my soapbox. And so the curriculum, the reason why the curriculum redesign hasn't been tested yet is because even though last year it was out, the APs were obviously went through a huge overhaul with the pandemic. So it wasn't tested in its original design and form. And so now that we have the opportunity to do that, and at least one paper administration, we will finally get a full test to be able to see what the curriculum is as it is expressed.
And now Melanie said that there's the first real full test. That's the first test that's being assessed on students. There are 3 practice exams that were created. Thank you, College Board, because they made 3 practice exams and not the other.
Do you think they're watching? What if they were?
I don't
watching?
know. Just saying.
Thank you, College Board.
Yes. Y'all should thank College Board too, because AP Chemistry didn't get new ones. I found out the other day when I was talking to a colleague, AP Chem didn't get practice exams. They're just like, oh, look at this old exam and look at this old exam. What? So they didn't actually get anything new. So thank you, College Board.
Is from like 97.
Yeah,
they're bad.
They're bad.
It's bad.
So anyways, but I was saying, so we have 3 exams that we can work with. So if your teacher has not planned for there to be a practice exam, say to your teacher, hey, you know, is there any way that we can do an after school session? Is there any way that we could take a practice exam during class? Is there any way we can do a Saturday session? Just ask them. The worst that they can tell you is no, right?
Yeah.
Or better yet, maybe they've already got it planned and they haven't told you that they have it planned. Some of us are very sneaky. Me in particular. And I don't tell my students what I'm doing. They don't even know what's coming on the pipeline to them. So it's possible that they probably are doing something.
And I have the exact opposite. I have our entire review schedule and all of our links open for them to look at for the rest of the time to the exam. Just
because things are open doesn't mean anyone's looking at them. Okay, so back to biology for our last 12 minutes.
Okay.
So what is the difference between sex-linked inheritance and autosomal inheritance? And how would you see the differently on a pedigree?
Ooh, good question. All right. So first of all, chromosomes, pair 23, sex-linked, 1 to 22, autosomal. So when you're working through an autosomal pattern, male affected, female affected, circles versus squares, it shouldn't give you a lot of information in terms of like proportion of who's affected. You want to focus more on, is it skipping generations? Do I see if I have an affected parent, are all offspring affected? That would tell you if you have something dominant versus recessive. So those are some pretty dead giveaways here. If you are going to look at something that is sex-linked, first and foremost, you should see something disproportionately that males are typically more affected. And you could have a skipping of generation. So for example, you may have 2 unaffected parents and an affected son. Okay, so that's going to show you that it could be sex-linked recessive because mom might have passed down that trait. But you're going to see this kind of ping-ponging around your pedigree and looking for patterns. My biggest advice to you, if you are stuck, take a guess and follow it through, right? If you really don't know where to start and the one you're most comfortable with autosomal dominant, but if it's autosomal dominant, it has to show up in every generation. You're not going to see it skipping if you have affected parents and offspring. All right. How would you notice a mitochondrial inheritance on a pedigree? It's not in this question, but.
Yeah. Your affected mother and all of the affected children. Because the fact that, and this is according to CD, the mitochondria and the chloroplast, which there has not been a chloroplast question about this. So I kind of feel like the chloroplast is probably going to get some love, maybe.
Yeah.
So the mitochondria or the chloroplast is going to be inherited through the ova or the egg. So it will always come from the mother. So
all of the offspring.
All of the offspring,
all
because the egg is where that mitochondria or the chloroplast is. The sperm is not going to have the mitochondria or the chloroplast in it. Thus, when those nuclei fuse, well, those cells fuse, like everything comes from the mother. So if there's a problem with the mother, then it's going to be in all of the offspring.
Mm-hmm.
Oh, you're
the
welcome.
reason why you see males tend to be more affected by sex-linked recessive is because they only contain one X chromosome, and they don't have an additional X to kind of cover up that affected X. They will have only Y.
This person really wants to know the answer, and so I figured I'd let you explain what you're talking about.
All right, so it's really lame, but like if you take your hand and you put it in front of your face and you breathe out, you feel the product of cellular respiration. It's so cheeky, but it works, right? So you have your gas, there's your carbon dioxide, you feel heat as an expression of your energy, even though it's ATP is what we're focusing on. And you feel your moisture. You got your water.
You get so many hearts. Like cellular every time you talk, there's like,
you're just not paying attention.
There's so many hearts that come up every time you talk. Me, I get one little heart that just floats up. And then you, it's like, heart. They
Samantha
love
was
you.
in our virtual classroom the other night. You're like all over the place. I love it, Samantha. Thanks for coming back over and over again.
Okay, we need
a couple more minutes. Look, you're getting hearts right now. There you go.
Yeah, that's because they're pity hearts. You know they're pity hearts. Don't play this game with me. I'm better. No
goodness. I'm gonna look in the CED real quick to see if there's something we did not mention.
Okay. Thank you, Miss Pratt.
We didn't talk about anything with meiosis. We should do that before this is over.
So random fertilization. Random fertilization has to do with that any sperm can fuse with any egg. It is random which of the eggs completes meiosis in the female to be released out as the egg. Thanks for my pity hearts. And then we also have that any sperm, because there's tons of sperm released, can fuse with that egg to allow for fertilization. So I think it's 72 trillion different combinations.
Yeah, it's absurd.
Yeah. Okay. So meiosis is very similar to mitosis, except for we're going to make gametes. And so although we start out with a diploid parent cell, the daughter cells are going to be haploid. So they'll only have one set of the chromosomes. So it goes through the same phases of prophase, metaphase, anaphase, and telophase. Remember, interphase happens before. Cytokinesis happens after. And you don't actually have to know those phases. That's pre-material. That's like material you would know before biology. But you have to be able to apply it. Also, if you do decide to give those words, make sure you use them correctly. If you use them incorrectly, you could lose points for bad. Oh, you don't lose points. You may not get your points because you have bad bio. So be very careful about using words you don't know. It's better to describe something sometimes than to throw the wrong word out there, especially if you're going to describe it. Like, oh, anaphase is when the chromosomes line up on the metaphase plate. What? You just said the wrong thing. So if you just said that in both processes they have anaphase, point. In both processes they have this process where the chromosomes line up on the metaphase plate, point. But the fact that you put those both together, you did not get that point. So be very careful on the way you word things. Prophase, you're preparing, you're kind of having your sister chromosomes or your homologous chromosomes are going to pair up together and line up. That's when the process of crossing over takes place, in which you can exchange genetic information and make recombinant chromosomes. Melanie, take it away with metaphase.
Yep, so our recombinant chromosomes are going to pair up as homologs or homologous pairs. And that's really important because we want to cut that chromosome number from 2n to n, right? Using the algebraic expression so encompassing all organisms. Homologous pairs line up and then they will pull apart because our spindle fibers are shortening and they're going to pull our homologous pairs to separate poles of the cell. We have now become haploid, but we have a problem because our chromosomes are still duplicated. And even though they are duplicated because we had crossing over, they are no longer identical sister chromatids. And that's another factor which increases our genetic diversity. Now we have two cells, still duplicated, and we go into meiosis 2. Meiosis 2 looks just like mitosis. We are going to increase the quantity that we have. So you're going to go through prophase, no crossing over because we don't have homologs anymore. Prophase, metaphase, anaphase, and telophase, pulling apart our centromeres, pulling apart our sister chromatids, and you are going to end up with independent chromosomes from your homologous pairs in those cells. Which homologous pair? Which piece of the homologous pair? That's another piece of randomness. But you should have one copy for humans of 1 to 23. Right.
And so, real quick, if the homologs don't move apart during this process, what is that called?
Dun, dun, dun. Different types of ploidy, right?
Oh, I was talking about one. Like if we have one of the homologs doesn't come apart, like I don't know, chromosome 22 or something. 21, sorry, chromosome 21 or something. If it doesn't come apart, that causes non-disjunction.
Non-disjunction. There you go.
Which can lead to a chromosomal disorder. And so what they can do is they can show you a karyotype And then they can ask you questions about that. And since there would be 3 copies of chromosome 21 after the sperm and egg came together, that could lead to Down syndrome.
A karyotype is a mapping of chromosomes on a like physically arranged from 1 to 23.
And you mentioned genetic variation, so the different ways to get genetic variation from meiosis is
mutation. Oh, sorry.
You're good. Mutations. But the reshuffling has to do with the crossing over, independent assortment, and then that random fertilization that started this whole process that we all started questioning. And you're right, mutation is how we can also get genetic diversity, and it's the only one that added new genetic diversity. Everything else that we mentioned is all reshuffling the diversity that's already there. So
and mutations get such a bad rap, but like, listen, mutations are not all bad. It's a spice of life. It's what made us who we are today.
We didn't talk about any of the mutations.
Well, or biotechnology, I just realized that. Biotechnology, look up some, go to Lab Exchange, watch some different processes happening. Looking at your PCR, which is really how all the COVID testing is so successful.
Oh my God.
Right? Go look at your gel electrophoresis. Go look at bacterial transformation.
It's a
big, 6 is big. Yeah. Application questions for a lot of that stuff. Because you technically don't have to know the specifics of how they work, but really the broader scope of what they can be used for and the information that you can get out of it.
Yeah, I think we got everything that we were going to answer in the chat, because the other stuff is applying to other units, or it applies to stuff we already answered, or it's asking questions about the exam. So I would like to remind everyone that you are a rock star, you're an AP Bio penguin, and you are dressed for success, or you are a recapper.
Yeah, or you're a recapper, which doesn't really suck, but that's okay.
I didn't think about that.
Yeah, you are in like the binge phase. Go for a walk, pop on some podcasts for the topics you feel less comfortable in. I know it can be really like affirming to keep going over the things you know, but you're wasting your time. If you know it today, you're going to know it in 3 to 4 weeks. Okay. Really dive into the parts of the curriculum you feel really uncomfortable about, because that's where you're going to get the most growth. So find the topics you don't like.
And when you are doing those practice exams, so if you have a Barron's book or any other test prep books out there, or if you're going through my Beast review guide and you see that you got something wrong, what you need to do is you need to focus on that, okay, and find out why you got it wrong. What misconception do you have that's causing you to get it wrong? Because a lot of the questions, multiple choice and free response, it's going to try to lead you to wrong answers. The multiple choice is going to have correct statements and incorrect statements in there, okay? And so those incorrect statements are designed to sound correct to somebody who actually doesn't know their content. So be sure that you understand why you got something wrong so that you can grow from this, okay? Those practice exams you're taking them, they're practice, okay? And it's meant to help you to get better. So don't just look at your score and go, oh, I got all these wrong. Good, I got these wrong. Because you know what? If you get it wrong because you guessed on it, if you get it right because you guessed on it, you're not going to go back and look at it. But if you get it wrong and you had no clue, that's okay, because then you can go back and figure out what you got wrong or what you don't understand. It is a growing process. And you know what? We're in a marathon right now, and we're almost in a sprint. Okay, there's 5 weeks left. Like
one of the things I would do for sure is like, because you just said something really important. If you guessed on it and got it right, but you won't grow from that. So here's my trick. Okay, use 3 different colors: green, and go through all the questions that you're so confident in. Okay, if you're not confident, answer it in like a yellow or an orange. Okay, and if you are guessing 100%, answer it in a red. Okay, because that's going to give you some sort of like triaging of where you think you are and matching it up to which ones you get wrong. So if you were getting a lot of green ones wrong, but you thought you knew it, like both feet were all the way in it, that's a real big disconnect for you. If you're getting a bunch of questions wrong, but they were the ones you marked as red, you're like, all right, well, I have a pretty good understanding of what I don't know. So now I can dive into that piece of it too. But you don't want to lead yourself astray by thinking you know something you may not. So that's definitely a strategy that can kind of like rope that into.
Okay. So remember, you got this. We have 5 weeks before the exam. We will see everybody next Sunday, to go over Unit 7. Yeah, like the next, I think in the next 5 weeks, we have 4 different sessions, because we had this one today. Unit 7 is next weekend. We have a bye weekend, and then we'll do Unit 8, and then we'll do the big review before the exam.
And listen, I'm sure we'll come back before administration 2 and 3. I have a feeling she'll
maybe rope me into it, or I'll rope her into it.
Someone will bring it up.
Know, we just do this because we like biology and we love you all. We get no money for this. This is just us having
100%
fun.
Free. Yeah. Last year when Miss Jones and I became friends through Instagram, it was like, hey, you want to go Instagram live together? I don't know how it works, but like, let's see what happens. Maybe someone will come. And here we are.
Oh, and P.S. if you don't know about HHMI, go do the HHMI activities. Those will help you review too.
That's true. They really will.
We're good. We're done, y'all.
All right. Have a good night, everybody. See you later.
Bye.